So that I really explained kind of the basis of my project there. Tonight we're going to talk about our summer. So we've been up here since June. This is Judith, Savoie.
Master today.
Yeah.
So I'm from Quebec, Rimouski. So I'm finishing my degree in chemistry. So that's my final internship project. I'm working on the metal, heavy metal in the salmon. So particularly the mercury.
So tonight we'll go through kind of where we caught the fish, how many we did, the numbers of adults we saw in subsistence fishery, and show you a few pictures of our summer. So we're going to go through those locations, talk about the par and the adults. We'll go through the kind of analysis, like what we're going to do in brief detail with fin clips for the genetic analysis and the otoliths that we collected. Go through a timeline, kind of give you what's the next year look like, what are we thinking of doing next year, and then we'll open it up for questions and we can talk about the salmon area. So my objectives for this project was to test to see if the salmon in Lake Melville are genetically distinct from the rest of the coast of Labrador, the rest of the North Atlantic. How do they fit in terms of relation to other populations of salmon? And to see if any populations within the lake are exhibiting any local adaptation, which means they are becoming more specialized to that river, which makes them more unique, and that'd be something to look into protecting maybe that population as more genetically unique than other populations. We also want to look at the subsistence fishery and determine if there is a mixed stock harvest happening or a single stock. So where are those fish that are being caught? Where are they coming from? Are they all originating out of Lake Melville? And then the other question is, where are they going? So what rivers are the majority of the fish that are being caught in subsistence fishery? What is making up the largest percentage of those fish? What rivers are the important ones? Is it Kenemu? Is it Cricket? Where are these fish going? Which will help to also identify those important rivers for protection for the future. So getting there. So luckily, the Labrador Institute has a couple of research vessels, and it worked out really nice. We used the 26-foot silver dolphin. I'm sure you saw me driving around town with it, and that fit in the flat-back canoe perfectly inside the big boat. And we had a little 2.5 horse kicker, so we would drive the big boat to the mouth of the river. We'd moor all of our gear into the flat-back canoe and head upriver until we got to suitable habitat. On rivers that were just too many kilometers upriver, we're talking 40, 50 kilometers to get up to where it looks to be good potential. We took the helicopter. We did that twice. We did that on the Red Wine and we did that on Mulligan. And we also used the smaller 18-foot crest lining that they have to do a few of the closer ones, Cape Caribou, as well as some on the Lower Churchill.
So some wildlife that we saw were great orange owl, storm merganser, so butterfly, moose, bear, black bear, stern, and sea duck.
And we saw a few bears throughout summer, but we didn't have any bad encounters. Most of them were pretty nice, and they were curious, but they kept their distance. So these are the rivers that we attempted to sample. So in all, it was 21 rivers that we ended up physically going to, on the ground, sampling, and trying to see if there were salmon there, what we were catching, brook trout, what it might be. So pretty good distribution. I've got Grand Lake, North Shore, got a lot in the west here in the Lower Churchill, and then a few on the east and the South Shore. So out of those group of 21 rivers, pretty much everywhere we went, brook trout are prevalent. You guys know that. You guys have been out fishing. They're everywhere. 11 of the rivers that we sampled out of 21, we successfully caught and captured our quota, our goal for the salmon. So still, we got a distribution. We got one in the east there, Main Brook. We'll talk about each individual river in a bit. And we captured a total of 531 par. So how do we get them? So when we're up in the rivers, normally when you capture juvenile fish, you use backpack electrofishing. You put a current in the water, it freezes them, and they float up and you net them. But the conductivity up here in Labrador that we ran into in the rivers, they weren't effectively getting shocked, and there was more just scaring them. So it turned out the most effective method was actually fly fishing. And something that I've done my entire life, and Judith is now new to the sport and picked it up really well. But it can be very effective. Some of the rivers, we catch our 50 fish in under an hour. And some other rivers, it took us 4 different trips to get 25. So every river had a different kind of relative ease of fishing and population size, quote unquote. But so we would fly fish while we're up there. We'd take a water sample, and we'd filter that. And then while we're in the field, we also weigh, we measure them. We take fin clip as well as the heart out of the par. And then we bring that par back into the lab, and we extract the otolith, which we were doing with the adults. You guys saw down at the dock if you witnessed Lana working down there. But in the par, they're so tiny that you have to go under the microscope in order to find them. Look like a grain of salt. So we'll start in the east. So this is Main Brook. I went over to Rigolette for a week and worked out of there and trained in the other New Nassau government hired Josh Adams, who worked on the subsistence fishery out of Rigolette. So trained him in, and Sam Palliser took me around and had him my guide for the week, and we ended up going to Main Brook as well as a couple other rivers in Rigolette, and that was the only one that we caught our fish, and it was fast, 45 minutes. We had our 50 fish in our 2 sites. We processed them. And it was good to see that in the lake. Totally different kind of ecosystems. All right, it's more barren landscape compared to Upper Grand Lake. It's totally different forest type. So along the north shore, right there, we go at Mulligan River, which I said we went on helicopter, and we went up to the split on the north branch and the west branch, and right at that confluence was right here. And so we sampled up north branch here, then downstream, which is flowing downstream out to Mulligan Bay, and our other site is down here. On the Savoie River, we went up the main stem to the first falls, which is about 7 kilometers up, and this is that set of rapids to the falls. And we caught, each river we tried to have 2 sites, right, so 25 in both locations. So we caught our 25 at this site, harbor fishing. It took us 4 hours or something. And then we went back another day, and there's a big branch off to the west, bigger tributary on Savoie. We went up that another 7-8 kilometers, and we fished that again for 4 hours, but we caught 5 fish. And so we caught a lot of brook trout, but not a lot of salmon. So where were those fish coming from? This side? Either way, it seemed to be a smaller population of salmon. It's a lot harder fishing on catch per unit effort, and we ended up with 30 total, which we can still work with for the project. Our original goal was obviously 50, but we couldn't get that. So in the west, we did Kenmu. We accessed that off the highway. We did the bridge site, and also there's a gravel pit on the east side. We can access the lower river there, and that was relatively easy fishing. Traverse Pond is another one we accessed by the truck, and we fished both the main stem below the waterfall on Traverse Pond there, and we only caught brook trout, no salmon. We fished the next branch, only brook trout, no salmon, and finally the farthest west branch is where we found salmon. So interesting, it has a number of factors. It could be waterfalls downstream of where we were fishing, so the salmon can't get up that far to where like the road is crossing. But we were able to find them on the farthest west branch. We also went to Caroline off the Lower Churchill. So if you cross the bridge going out towards the coast and you look upstream on the Churchill, it's the first river that comes in. There's a nice green spot you can see. Went up there, and there's only about a 300-meter section of suitable habitat for the par before it gets to a series of waterfalls that's impassable. It's a very small section of quality habitat, and it took us 4 trips to get 25 par out of that site. So much smaller population, relative population. And then we fished Peters, which is a tributary of the Goose, and we fished that at the base of Peters Falls was one of our sites, and then at the confluence of Peter's and the Goose, there's another good set of rapids there. So that's where we had our sites. And Upper Grand Lake, we had Crooked River, which is a beautiful river to go up on. Easy fishing, and this was my personal smallest, best fish of the summer. It was 66 millimeters on fly rod. I don't know if that's some sort of record, but we also went up Redwine. That was helicopter access, and we ended up way up on the headwaters of Redwine, and that was very easy fishing as well. The Susan, we accessed that by boat here. This is going up into the mouth of Susan there. And went up a ways, caught our fish fairly easy. Good number of them around. And then Cape Caribou was the last one. And I'm going to talk a little bit more about Cape Caribou. We have 76 par out of there, and I'll tell you why. Originally, our two sites were off of the Grand Lake access road. And so the first site was at the bridge crossing. This is the main stem of Cape Caribou here. And so we caught our 26 up there, and then off Alderbrook, at this bridge crossing, we caught another 25. So that was our original, okay, we have our 50 for the river. And after talking to a few people around town, it's kind of come up that there possibly this could be a one on each population because of some falls that are occurred downstream of this site. So looking on Google Earth, you can see this is the first big set of falls. This is at River Founder 12, and this, according to the literature that I could read in the Rivers of Labrador, is saying that this is probably a complete barrier to upstream fish passage. And if you look at the downstream sections, there's the mouth, all that white water, there's 7 other falls downstream of this major one that could be impeding that movement. So the fish we sampled up above, those could be a Wananese landlocked population. So we went back in and we sampled the lower stream section and got an additional 25. So we'll be able to compare those downstream fish to the upstream fish, see if there's any difference. And if I'm hoping the lineup and the weather cooperates, we're going to come back and try to we're going to hire a guy in town that has a plane, and we're going to go get a visual on this waterfall to really see what's that vertical drop. Is it a complete barrier or is it a partial barrier? So this is kind of in the works, but that's why we have more fish on Cape Caribou. So this is all of the salmon and their lengths there. And so we had 531. We averaged about 114 millimeters, which is not bad. Our smallest one was 42. That was e-fishing. And our largest one was 170. But you can see just nice bell curve distribution from 40 into this is our kind of sweet spot from 100 to 130 millimeters of where majority of our fish were, and one up into the 170. If we look at, since so far, the only analysis we could do is just based off of the data we collected, which was weights and lengths. So this is a body condition index. It's called Fulton's K. And it's an easy way to see if your fish is short and plump, is it long and skinny, it gives you an idea of the body condition of the health, right? Is it emaciated, is it too skinny and long, or is it overweight, is it feeding really well? And across all of our samples, we had an average of 0.97, so 1 would be a perfect weight to length ratio, nice average fish. And so this is pretty good to, we have healthy fish in the river. There's no really skinny long ones, which would be an indicator of lesser health. And this is plotted around, this starts with the smallest fish and works up to the largest par that we have. And you can see there's a trend line in here, and it's slightly decreasing, so it shows that the younger fish in the river are a little more plump and fat, and as they get older, they're kind of getting skinnier and maturing. But it's good that the younger fish they're eating obviously very well if they have higher weight per length. So this is good. So we have healthy par in all the rivers that we saw. This is a fishability index. This is something we've talked, this is our opinion on there's a definite relationship between catch per unit effort and relative to population size. So the ease of fishing and how fast we got our samples is probably directly correlated to potential population size and will really identify like the bigger producers of salmon in the lake. So this is excluding Kenemu, because Kenemu is just on a totally different scale of all the other rivers that we sampled. So our easiest ones, the ones that we were in and out way faster than we thought we would be, were Mulligan, Red Wine, Mainbrook, and Crooked. So those were really the fastest ones we could do. Traverse Pine, Susan Peters were a little harder to get our samples, but Cape Caribou, the downstream section, we fished really hard to get those 25. Caroline took us 4 trips to get our 25, so it's directly relating that you could go on a limb. This is just my opinion. We didn't do a population assessment, but based off this catch per unit effort, you could say that these rivers are having a less of a contribution to the number of salmon that are present going through Lake Melville and Grand Lake compared to these. So you got 2 upper in Grand Lake, you got Mainbrook on the east, and you got Mulligan on the north, and obviously Kenemu on the south shore. So those are really those are the big rivers from this standpoint. This analysis was just what we saw in the field, and when we do the genetic work and the otolith stuff, we'll really be able to confirm that. But I wanted to kind of go out and say those I could probably say is going to come back as being the big players of the rivers producing salmon in Lake Melville. So Judith will talk a little bit about the adults.
So we sampled the adult salmon from the subsistence fishery here in Northwest River and in Rigolet. Here in Northwest River it was Leona Rice, and in Rigolet it was John Adams. So what we also sampled the fish from the community freezer here in Northwest River. And what we sampled on the adult fish was a film clip for the genetic and also the otolith that's situated just under the brain here. And for the metal analysis, we sampled the operculate and a meat sample just right here. So when we were in the river, we caught some mature par that here on that picture, 3 mature par. And that's par that developed sexual characteristic, but never left the river, so they didn't went to the ocean. And we also caught 1 grills, and that's an adult that left the river for only a summer. So usually a salmon left the river for a year and went to the ocean for a year, but he just left for a summer. So we sampled this summer 424 adult from the subsistence fishery at 31 different locations. So the maps show all the different locations we collect the adult, and we clearly see 2 hotspots, one in the mouth of the Kenemu and Kennebec River, and one in Rigolet. So the 3 major par that we caught was 2 in the Peters River, again, and 1 in the English River. This one is really interesting because in the English River, it's the only fish we caught there. So we are wondering if the fish is coming from a landlocked population over the waterfall and he went downstream, or if he was just lost. Or we heard too that there is another side channel on the English River that maybe the fish are going there and he was on the main channel. So that's something that the genetic and the microchemistry from the otolith will have to answer the question. Yeah, and the grills was caught in the Crooked River right here. So we sampled 282 fish caught in the area of Northwest River, and the average length was 58.2 centimeter, and the largest one 91.1, and the smallest one 21.3. And there's not a significant difference between the one we caught in the area of Rigolet, because the average length is 56.7, and the largest one is close to the other one, 90 centimeters. And the smallest one is a little bigger. 24.1 centimeters. On that picture, we clearly see that is all the fish from Northwest River and Rigolet together. So we clearly see that the majority average length of the fish was right here, and that corresponds to the length of the first year spawner. That is normal to have in bigger quantity.
Yeah, so the majority of being in that range, that's the size of the salmon that would be returning for their first spawning attempt, right? So Atlantic salmon can spawn multiple times over many years, but majority of fish don't come back. So what happens is they don't survive that initial spawning, but you do get a group that will go back out for another season. They grow bigger and they come back. So this is where you start to see those next bigger fish, right? So you get over those 80, 90 centimeter fish. That's normally your second return spawners. So majority of the fish that we sampled are the first year spawning salmon. So standard size coming back in. We didn't see a lot of big fish, but maybe we just didn't sample during that time of year, or everyone was tagged out before the big fish ran, right? So there's a lot of this could be sampling bias, but for what we saw, a majority first year spawners.
So I have 130 adult sample for the middle concentration analysis. That is the meat sample and the operculum sample. That's from the adult. And I have 456 opercula from the par. So the meat sample will be used to determine the concentration in heavy metal, particularly the mercury, because we know that it's a health preoccupation, but also nickel, lead, arsenic, and really a lot of metal we can have. So and also that will give us a data from concentration before there's muskrat fall build, finish to build, and in operational, and before mines. So if in the future we are seeing an increasing concentration in the fish, we'll know there's a new source of contamination of the water. So we'll be able to relate that to the maybe muskrat fall or a mine. We'll use the adult opercula to develop a new method of analysis to know the concentration of heavy metal. We'll analysis like with the meat concentration and the concentration in the opercula. We'll try to find a ratio between them, and after that, in the future, we won't need the meat sample. Yeah, the meat sample. So the opercula is really easier to sample, and also it's not eatable. Yeah. So after that method is developed, I can analyze all the opercula from the par, and that will give me a general portrait of the concentration of heavy metal and metal in each different river, and if the small par are already contaminated by the concentration of mercury or nickel. Yeah. So when I'll be back in Rimouski, the first step I'll do is to analyze the meat sample, so knowing the concentration, mercury, nickel, lead, and whatever. And after, I'll also analyze my opercula. In January, I'll start my master at the Memorial University, and I'll work on the data I will receive from the analysis on the meat and the opercula. So I'll work to have a mercury concentration data, and also to find the ratio between the meat and the opercula. And after that, If everything goes well, next summer I'll do the analysis of the power porcula and maybe a field work. Yeah.
And for the genetic analysis, I'll be bringing the tissue samples that we collected, all the fin clips and the heart clips. We'll bring those down to Dalhousie in Halifax, and sort of my other advisor, Ian Bradbury, is working down there. And I'll head down there, extract the DNA, and prep it to send out to the Genome Center in Quebec. And we'll be doing this next generation RAD sequencing, which is one of the newest techniques to come out. And we look at the entire genome, and we can pull out these SNPs. So these are just in the genes. You have all these base pairs, and you look for these single differences in these similar sections. So in a particular river, with breeding and local adaptation, you would get a change in that similar section. So if you look at the section of the same section of the genome across all fish and all populations, similar fish that group together would have that same polymorphism. They might group differently from other ones. So you're just looking at kind of a fine scale resolution and comparing the genetic material of all these populations. And we can look at functional sections of the genome, which means that these can be acted on by the environment. So if river length might be a determining factor on the success of a salmon spawning. So if it's better fit for that particular physical characteristic of the river, it will pass on its genes, and you'll get a trait that is better suited for long distance travel upriver or high current or something. So you actually look at physical parameters on the landscape and tie it into the effect on the genome. And we'll be using a whole host of analytical programs to compare this SNP data. You get back tons and tons of files of data, so it's really hard to sift through and organize it effectively. And we're just going to be looking for that genetic structure. So how do the populations, 11 rivers that we sampled, how do they tie together? And then how do they fit into the larger picture of Labrador, North Atlantic? How do they, where do they fit in on a genetic scale? And we'll also be looking at the adults that we caught in subsistence fishing, doing that mixed stock analysis, kind of where are they coming from? We're not picking up any fish from New Brunswick or anything. Are they all originating from Lake Melville? For the otoliths, the way this works, I'll be doing this work this fall at 1, and we do laser ablation, and you can look at the different levels of stable isotopes in the chemistry of the bone. And so it's like a tree that puts on growth rings every year, so you can age the fish really well, so we'll be able to know how old the adults are. But also we'll be able to kind of key into the center core, and these are the layers that the adult put on when it was a juvenile in the river. So when the river is going through the landscape, they pick up this different water chemistry signature. The juvenile salmon in the river are taking that chemistry into their bones. This is one of the bony structures. And so we can kind of take the bone and the water samples that we collected and kind of get a river signature. Kind of Upper Grand Lake looks like this, or maybe the rivers will be individual, depending on the underlying geology. You could have varying levels of scale, but you should at least be able to determine Upper Grand Lake, South Shore, North Shore of Lake Melville, and then you can apply that to the adults and look at that intersection and reassign that similar river signature to a particular river. So you'll be able to reassign the adults. So these two, the genetic and the otolith, are kind of two independent tests that will kind of answer similar questions, and they should be used to confirm and back up each other. So the results should kind of come together with both of those techniques, and we'll be able to say, yes, this is where this salmon came from. If the genetics aren't different, then the otolith water chemistry stuff might be different. So we'll still be able to reassign, or so hopefully everything will be in agreement, and we'll be able to reassign these adults. So timeline, so this fall, I'll be working on the otolith stuff in St. John's, and then going down to Halifax to work on the genetic component. Winter, I'll be doing analysis of hopefully the otolith data. I'll be able to get that done fairly soon. The genetic stuff takes a while to send out. And hopefully we'll come back, whether it's this fall, late this fall, or in the spring for sure, we'll come back and we'll give a preliminary results on at least the otolith side and some of your...
I hope my meat harvest will be done, so maybe I'll have some result in the mercury level.
Yeah. So hopefully we'll come back up and let you guys know kind of what we found in the beginning. And then next summer, since we did so well, this was supposed to take 2 summers to get we wanted 10 rivers and around 400 adults, and we got 11 rivers and 424, so we're ahead of schedule. So now we can kind of look outside Lake Melville, and there's a lack of information from Rigolette north. So there's a lot of rivers that have not been sampled by like DFO for genetic work. So there's been 4 done, but there's a lot more that have potential. So we're going to look into the feasibility of at least getting outside Rigolette and Dublmer and start from there and try to get increase the information available for northern Labrador, because it's a spot that's lacking for Atlantic salmon. So in summary, we went very successful summer. It was great to be up here for the 2.5 months that we were here. And we got to see a lot of the country and a lot of the rivers. We lucked out, I think, on bugs. We were on water so much, it was pretty good. We picked windy days to work. There's only a few nights that were pretty bad in the tent. But we got 531 salmon par from 11 rivers and 424 adults from those 31 locations, which is spread throughout the lake, which is really nice as well. So we'll be able to tell where that adult was caught and where it was headed to. We're doing this combination of genetic as well as microchemistry analysis, and we're looking at that structure, how everything ties in the bigger picture. But the main thing is also identifying those important rivers to the subsistence fishery and to the area, so you know what rivers are producing more percentage of the fish caught in the subsistence fishery, so you know those are important rivers to protect for future generations if you want continuing healthy runs of salmon in Lake Melville. A lot of partners and funding groups that helped this summer, and I'd like to thank my advisors, Dr. Marie Clement and Dr. Ian Bradbury, as well as my other committee members. I'd like to thank the Natsui government and the Innu Nation and all the community members in Northwest River, Shashashi, and Rigolette. You guys were very helpful in letting us take samples off your salmon, and everyone seemed very interested in the project and helped me out with tips on navigating different rivers and where to park and where to do all that stuff. So it was very community kind of came forward and really helped us out a lot. So really appreciate that, and I'd like to thank our 2 technicians, Leander Rice and Josh Adams, for their hard work this summer. With that, we'll field some questions. So. No questions, there's food.
Is this the first time that something with regard to the study in this nature had been done?
In the area, this technique of using genetics and otolith has been published before in other areas and other species. For Lake Melville, though, and in Labrador, the most recent information and kind of project that happened was a few years ago. My advisor Ian Bradbury, with DFO, collected samples from 23 rivers across southern Labrador, 4 in Lake Melville, and 4 north of Lake Melville. And that data, they used an older genetic technique called microsatellites, and that looks at non-coding sections of the genome. And I worked up that data this winter, and I do have it, so we wanted to pause and I could show you that data as well. And that shows that Lake Melville does separate with that data, not significantly, but it's a trend. And with microsatellites, normally what happens is if you can start to see something with microsatellites, it's a less fine technique. With this new next-gen sequencing, we should be able to distinguish those populations. But Lake Melville did group away from the rest of coastal Labrador, and there was also a few rivers in southern Labrador as well as in northern Labrador that separated out as well from kind of a cluster around like Cartwright and down. There's a lot of rivers in there, and we can go over that data if you want in a bit. But as far as Lake Melville, yeah, we've just learned with this, we have a fine scale resolution, what's happening right within the lake. We went to almost every major river that is flowing into Grand Lake or Lake Melville. So we found the salmon where they're present. There's a few rivers I'd like to check out maybe next year. But we covered our basis up here, so we should be able to have a pretty fine scale results for local conditions.
How do you determine adult salmon?
When it's a juvenile?
The adult.
Oh, okay. Like the mature par that we found?
Like bones and size?
Oh, normally, so the par will stay in the river 2-3 years. They have those par marks, right? So they get to a certain size, and they out-migrate to the ocean. They switch food sources. They grow larger. They have that chrome color change because they need to adapt to the ocean environment, which blackback silver chrome size helps with camouflage in the water column. So normally that's the determining factor. Second, they leave the river, they change their coloration, and they get a lot larger. With the mature par that we caught, the way you tell those is kind of know, we'll take a bit to get there, but so you kind of know roughly what size they should be leaving the river, which is around that 170, 180 millimeter size. Is normally get to that too large for, if that par was going to leave, they should head out, right? And so when we catch a par in the river that's 200, 210, and we sample it and you cut them open, actually most of the time they're males, because males will, it's a life history technique that they have, that they just say, I don't need to get big, I'm just going to get my sperm sacs ready and be ready for the fall spawn. So they're like, I'm not going to leave. They don't make that choice, but somehow they determine, I'm just going to stay here. This is fun. I don't need to go to the ocean to get big. And they do contribute to fall spawning, these small fish. And so anyways, so normally there's a clear size restriction. The fish leave, the par leave, and they get really big. But you get some large par that stay behind. And they have mature sexual organs and they're ready to spawn. And the grills is just, it's purely based on size. I think that one was 220 millimeters that we got up there. And there's a few other fish from the subsistence fisheries, those that grills. But he was chromed, so his coloration showed that he went to the ocean. He came back, so he had that chrome color. Where this fish, although he's almost the same size as this fish, still has par bars on him and has that dark coloration which you find in the river. So it's just a different life history trait, and they kind of hang out and wait for the big males and females to get together, and they come in and contribute. You know, they can't fight for a spot in the river to spawn with a female, but they can get in there real quick.
How far up the Nisgabi were you?
The farthest tributary up was the Red Line. I looked into the possibilities of getting up like the North Pole and getting up there, but other than that, when we accessed it, yeah, we could almost go there. But we went up helicopter there because it was, almost a 60-kilometer run up Grand Lake, and then another 20, 30 up the Noscopy to the mouth, and then it would have been 40 up to where we wanted to go. So that was just taking too many days. So the farthest we went up from the bottom was just the mouth of Crooked, and then went up Crooked with the canoe.
Did you find any salmon in there?
In here, in the Crooked?
there's a lake in there they call Salmon Lake
Yeah.
before you get to, what's the lake? Seal Lake.
Yeah.
Outside of Seal Lake. There's a Salmon Lake that they call it. The salmon's supposed to go there and spawn.
Yeah, I know they go up the Nascapi up to where they call it the gorge. I think it's a serious set of rapids up there, and according to like Rivers of Labrador, I've obviously never physically seen what those rapids look like, but from the description, they think it's pretty impassable and be really tough for them. But there's not to say that there isn't like a Wanadish population up in the lakes, right? So there's a lot of that where at different, when the sea levels were higher, back when glaciation was receding and these Atlantic salmon were recolonizing the coastline, they could get in past these rapids didn't exist, right, at the time. So they got up above them, sea levels dropped, they got isolated. So there's definitely a landlocked Wanadish populations around. But we didn't get far enough up here. There's a few tributaries I wanted to look at, but that's always helicopter time, and it gets very expensive very fast.
Far as what happened in Mulligan River was to the branch there, was it?
Yeah, so the
Right.
split, yeah. And then there's a serious set of rapids there and falls. And that's pretty much, from my understanding, they don't think they can get past there.
No.
I did see them when we were flying in. You can look at it, and there's a lot of white water. So we got pretty much up to where I think we sampled up where they were, and they were there. I mean, it was, you couldn't keep the fly in the water without catching them. So.
Do you have any thoughts on why Savoie didn't have as many salmon?
Yeah, so from what I've heard is that there was a big fire. I don't really know my dates, but I think in the 80s possibly there was a big fire up in this river basin here. And with fire, you get increased sedimentation in the river, which messes with spawning and egg development, but also you lose your canopy cover, right? So you lose all the trees that were shading the river, so you get increased water temperature combined with the increased silt. Could have kind of hurt, taken the population down a bit. It looks like it's like good habitat on this west branch here. I don't know the actual name. I don't know if you guys do, but yeah. But we went up and we got to this beautiful section that was like 2 kilometers long of riffles and nice pools, and there's 5 salmon, but there should have been salmon there, and it was just brook trout. And that section, if they can't get above that set of rapids on the main stem of Savoie, then it's a really short section also for habitat-wise. It just wouldn't be able to support big population either. If they could get up through the rapids and those falls, I did look at it. They're not huge jumps, but there's a good amount of current going in, especially the first couple aren't bad, but the highest one is pretty substantial. And then it's a long way up after that set of rapids to where it starts to get to look like good habitat again. So it could be a combination of that fire hurting it, and also could be a lack of adequate habitat.
You think that fire would have did the same in the Mulligan River, too?
Yeah, I think there's, like I said, there could be also that other factor of just lack of habitat. If they can't access above, that's really restricted then. That below the falls is only 150 meters worth of quality river, and then compared that to what we saw in Mulligan, the whole thing just looks beautiful. Like it's perfect salmon habitat. All right. Well, I appreciate you guys coming out, and yeah, we can talk if you want to see that genetic data. I have it right on the computer, so we can look at that if you want. If not, you can take a break and we can keep talking about the salmon rivers. But it was it's enjoyable to see all those areas for sure. So,
We enjoy our salmon.
yeah. Thank you.
Thank you.
Great.